So we'll get to that right now. Okay, go take it away, Tuval.
Great, thanks, Brianna. Before Brianna starts, I'll just give a quick overview of the technology, of some of the technology that Brianna used in her research. So Brianna used a bunch of different methodologies. One of them was DNA sequencing in order to identify, classify, and quantify microbial populations. What was unique about the technology that Brianna used specifically in her sequencing work is that she employed the use of long-read sequencing for determining microbial composition instead of the traditional short-read sequencing. So what I'll spend the next few minutes doing is just explain the basics of the long-read technology that was used in this work, and then I'll hand it back over to Brianna to talk about her exciting work about Kefir sequencing. Okay, so LoopSeq is a technology that essentially transforms short-read sequencing from Illumina into long-read sequencing. So it's a combination of chemistry and software that takes the existing short-read sequencer from Illumina and transforms it into a long-read sequencer. And this slide here basically explains how we do that. So starting from the very top, where it says attach, every LoopSeq sample preparation procedure starts by attaching a UMI, a unique molecular identifier, to every long DNA molecule. So in this slide here, that's that blue barcode-looking element on the DNA molecule on the top. That contains a UMI, which is a unique molecular identifier. A unique molecular identifier is simply a sequence of DNA that provides a unique address written in DNA sequence to each DNA molecule in the sample. So imagine that on the top there, we have full-length 1500 base pair V1, V9, 16S molecules, and we're attaching one of these UMIs, these unique addresses written in DNA for each and every molecule in the sample. Then we go through a step called the amplify step, the amplification step, where we create multiple copies of each barcoded molecule. And then we arrive at kind of what is the heart of our chemistry, which is the distribution step. In the distribution step, what we're doing using a series of enzymatic reactions is we're taking that UMI element, that DNA barcode, that molecular identifier, and we're copying and pasting it into multiple positions within the same molecule, but not to neighboring molecules. So it's an intramolecular barcode copy-paste operation. So now this information about the clonal origins of that molecule, the UMI information, is spread out throughout the length of the 1500 base pair fragment. And then in the final step, we take these long molecules that have the barcodes in them, and we fragment them down to short molecules that are short enough to be sequenced with any Illumina sequencer. So using, say, paired in 150 sequencing. And those are those shorter reads that you see next to sequence that have the barcodes on them. So imagine that what you're seeing here is a cluster of short reads that share the same barcode. That means that they originate from the same original DNA molecule. What our software then does is it takes the short reads that come off of the sequencer and it clusters them by their barcode, by their UMI. So every cluster of short reads that share the same UMI is then used in order to de novo assemble the full-length 16S molecule that UMI originally barcoded. So if we're, for example, if we tagged 10,000 full-length 16S molecules with UMIs, we will go through de novo assembly 10,000 times, each time with a different set of short reads that share the same barcode. And the long reads that are generated from that de novo assembly are written into FASTA and FASTQ files, which are the raw long read data. And that raw long read data is the data that Brianna used in order to classify and quantify the microbial composition in her samples. I should mention that this is a specific application of LoopSeq technology to full-length single molecule long read 16S sequencing, but the exact same chemistry can and is used to sequence any long molecule, DNA or RNA. And we have multiple applications for sequencing different types of amplicons from the microbiome and not from the microbiome, sequencing genomes and transcriptomes. So this is a general purpose long read chemistry that can be used to sequence any long DNA or RNA molecule. And the presentation that Brianna is going to present now is showing one very interesting application of using this technology for 16S sequencing for characterizing dairy products. So Brianna, take it away.
Thanks, Tuval. All right, so we'll go back to thank you everyone for being here today, and thank you Tuval for that introduction into the technology. We're really excited to use it and show you our results today. So today we're going to go over a presentation called the Pet and Human Commercial Kefir Products Assessed for Microbial Species Accuracy and Density. And we have a quick picture of one of our publications from the Journal of Animal Nutrition. So an overview of kefir is that it's a fermented food. Well, fermented foods and beverages have becoming more and more popular. You'll see yogurt, kimchi, kefir, sauerkraut, kombucha as well in the stores now, labeled as fermented and probiotic. And kefir is one of them. It's basically a thicker yogurt, and it's made by fermenting kefir grains in water or yeast. And the top 5 USA kefir producers generated over $85 million in sales in 2019, and the market consumer average growth rate is expected to be 5.8% in the next 5 years. So it's definitely an up-and-coming market, definitely worth the attention. Previous research in animal studies is sparse in terms of kefir. We have a lot more with humans. One of the 2 publications currently out that studied kefir effects in animals did show that dogs that were supplemented kefir for 2 weeks had an increase in Prevotellinaceae bacteria, Salmonellomonadaceae bacteria, and Sutterell-Aceae bacteria, and there was a decrease in Clostridiae, Fusoactinariae, and Romanococcaceae. And this is when kefir was consumed over 2 weeks, modified the gut microbiome, but there was no clinically adverse effects to the animals. And then we did see another study that tested in dogs. They were given Lactobacillus kefiri, which is a specific bacterial strain found in kefir. And this was also found to not influence the concentration of immunoglobulin A secretion. So overall, there were not any large significant changes to the animal's microbiome when given kefir supplements. In terms of human kefir research, we have a lot more data from small clinical trials to in vivo as well. So we have seen there's antibacterial effects to consuming kefir in terms of deleterious or bad bacteria. There is better blood glucose control after long-term kefir consumption, better blood cholesterol control, and also anti-inflammatory and antihypertensive activities noticed when taken regularly. There are a lot of potential pitfalls in this fermented foods market at the moment. The regulatory environment allows for a lot of areas for misreporting in terms of health claims, in terms of ingredients, or in terms of viable information on the label. And so the objective of our study was to measure the viable bacterial counts and characterize the bacterial taxa of commercial kefir products intended for companion animal consumption and also human consumption. And we did this by comparing the enumeration and taxonomy data to label claims made by each company. And our hypothesis was that the pet kefir products would not closely match label claims of viable colony counts or bacterial taxonomy, while human kefir products will be comparatively more accurate in the label claims. This is a quick graphical abstract of what we did in terms of methods. So for example, we had the 5 pet kefir products, and then we plated both. We had 2 lots of each product that we plated on basic MRS media, and then we were allowed those bacterial plates to grow for 48 hours before we measured their density. Then on the same day of opening, we sequenced the DNA that we extracted and submitted that to Loop Genomics to get a full sequence of what was in each product. The products were then tested again 14 days later for their microbial density to see if everything died off or if we had an increase in growth. And then finally, the products were evaluated if they met criteria or not. So for the 6 kefir pet products we used, we had product A, which was Honest Kitchen Daily Boosters Instant Goat Milk with probiotics for dogs and cats. We see that's the purple one on the bottom. And then product B was Open Farm Raw Organic Grass-Fed Kefir. Product C was Answers Pet Food Raw Cow's Milk Kefir. Product D was Coco Love Coconut Kefir for dog nuggets. Product E is Champion Choice Natural Kefir, which is the last tall bottle. Product F is not listed here as a picture, but that was only natural raw goat milk kefir. And our final was our control, or what we used to validate techniques, was Chobani's plain yogurt with 0% milk fat. So we figured that was a large generic yogurt brand that would serve as a pretty strong control for our experiments. For the human kefir products, we also did 2 lots of each. Listed below, we have product A, which is Maple Hill plain kefir. Product B was Siggy's dairy film jolk. Product C was Redwood Hills Farm Goat milk kefir. Product D was Koyo kefir, which is a product of GTS or kombucha. Product E was Lifeway Traditional Original Kefir, which is probably the most predominant kefir producer at the moment. And then as finally, again, our control was the Chobani plain yogurt. To enumerate the bacteria, we took 1 gram of each product and vortexed it with PBS, and then the combination was serially diluted on plates from 10 to the 5th up to 10 to the 7th, and they were plated via spiral plater so we could ensure consistent plating throughout each dilution. They were on MRS media, which is a traditional full nutrient media, and they were grown at aerobically at 30 degrees and anaerobically at 37 for 48 hours. So that was to ensure we could culture as many lactic acid bacteria as well as yeast bacteria as well. And the media, at the end of the culturing period, media with less than 20 colonies were discarded, and ones with more than 20 colonies were retained for analysis. And the data you'll see is presented by colony forming units per gram. So how many active spores were in that sample. To extract the DNA, we used a basic DNA Easy Powerlysal soil kit, and then we tested the quality of DNA on qubit and the quantity as well, the gel agarose. So everything checked out. And then as Tuval had mentioned before, we submitted the DNA for 16S long-read sequencing by Loop Genomics. And this was processed through Silva's 132 database. And more recently, we have an update from the Silva 138 database, which we have already run the data through again with some small updates. So this is a table representing all the pet products, how much bacteria they claimed on their label would be in their product. And then we have the results from the highest count from day 1 and the highest counts from day 14. So we're trying to give them their best shot here. And as you can see on the far right of the screen, we have a red X representing if it did not match label claims, and we have a green check mark to represent if it did match. So as you can see, all of these pet products, some of them got close, like 10 to the 8th, 10 to the 9, but none of these, as you can see, matched their label claims or surpassed it from our testing, except for the control, which is the yogurt, the Chobani, and that one did surpass, which did give us confidence that our methods were sound in that sense. And then we'll go into the pet products in detail. So this is the results of the loop sequencing. Things that are labeled in green means that they were listed on the Kefir label and were also detected by loop sequencing. Products in red mean that they were not listed on the label but picked up by loop. And the products listed in blue were listed on their label but not detected at all with our 16S sequencing. So here in product A, we see a majority, the top 4 bacteria were claimed on their label, which is great. And the most predominant was definitely Bacillus coagulans and then Lactobacillus plantarum. We also see several other species that were not claimed, but still had a good enough abundance that should be considered. For the next product, we have product B. This is the open farm kefir. This one, the top 3 abundant bacterial species were not listed on their label, but we do have the Lactobacillus debrucki, Lactobacillus casei were claimed, but you can see those are only around 4 to 5% of the total content. So, these might not be. The other ones are a lot more predominant and should definitely be listed. And you can also notice at the bottom, there's a good amount that were not listed, were not found in the product, but listed on the label, which would be Leuconostoc, Enterococcus, Bifidobacterium. Many of those people will buy in confidence, hoping that that's in that product. Product C was answers. Again, we have Lactococcus, and based on our new update, the most predominant species there was Lactococcus lactis cremoris. And then we also have Lactococcus lactis, and again, many products that were not claimed but were detected. And just in case there's confusion, the outer circles on each of these pictures, it represents one lot, and then the inner circle represents the second lot. So this is just an image of both, but these final numbers are averages from the two. Our next product is Cocoa Nuggets. This one did not claim any bacteria, so you will see all of these are in blue. They were most predominant in Streptococcus, Thermophilus, and then Lactobacillus spp. And then the next one, we have Champion's Choice, which is a local kefir brand in Illinois. And their two most predominant species were not listed on the label. And those, according to Silva 138, is also Lactococcus lactis subset Cremoris. And you can see these last four, Aciobacter, Lactobacillus cossicus, Leuconostoc, and Saccharomyces kefir. None of those were found via sequencing, but those were the most predominant listed on their label. And then the last pet product, we have Only Natural Pet. They did not claim any bacteria, so that's why these are all in blue as well. But you can see the predominant species here are Lactococcus, and then we do have some Citrobacter frondundi, but a very small amount, but was found in both lots, so still significant. And this is also the only product that we have listed that we actually detected Lactobacillus kefirano-facens, which is the most specific species to kefir products. So that was an interesting note. And then these are the results from our Chobani sequencing. Only one lot worked through sequencing. That's why you see one circle. But for the most part, they were right on the money with their Streptococcus. But with the Streptococcus salivarius and Lactobacillus paracasi, Lactobacillus ramus, those are not listed on the label, but they were detected in sequencing, so that could be down the road leading to more consequence. But for the most part, this one really validated our techniques for sequencing. So these are the results for, the human product cell count. So as you can see, we have the products listed on the far left, and then the claims that they had posted on their label next. Then we have the highest amount detected on day 1, highest amount detected on day 14, and then the final result is if it passes or does not pass. So the first two did not claim any CFU per gram on their label, but they did have the first one, product A, reached a max of 1 times 10 to the 9th, and then on day 14 had a max of 10 to the 10th. Product B maxed out at 10 to the 8th on both days. And then as you can see, product C, D, and E, none of those matched or surpassed their label claims from our results. And then again, the yogurt data is repeated, is not changed. So you can see that checked out. So we'll go into some more detail into the human sequencing, our human product sequencing. So in this Maple Hill, the most predominant strain was Streptococcus thermophilus, and then Streptococcus salivarius, Lactobacillus acidophilus. And I would just like to emphasize again that the way we were only able to get this specific to species due to the long read sequencing. So this is really exciting and new that we know the species level in some of these commercial products. Again, to note, there's no Leuconostoc found and no Bifidobacterium found in this product. The next one was Siggy's, which I'm sure you guys have seen in the store. I know I have, probably one of my favorites tasting wise. Again, their top 3 were claimed, so that was a lot more accurate, which is great. And again, we have a good handful, though, of species that were found via loop, but were not claimed on the label. And then our last 2 products were listed on the label, but not detected. So the new updated sequencing, could actually have some of that information. Then the next product, we have product C, Redwood Hill Farm. Again, top product or top species was Streptococcus thermophilus. And then a lot of green down the way, and we still don't have any Leuconostoc or Bifidobacterium found in these human products. So that is of note. And a lot of lot-to-lot variation, as you can see here. All right, so moving forward, we have Koyo. It's like a coconut milk kefir, so dairy-free, but still uses the fermented process. They claim billions of live probiotics. And so again, this is our next human product. We have a highest of Streptococcus thermophilus. So it seems like when you buy kefir for humans, you're most likely, that's what you're drinking the most of. In this product of note, we did detect Bifidobacterium and Lucana stock. So for the other products that claim these strains and we did not find them, this was our validation that, Loop and the databases were ready to receive it and definitely identified those species. And then the last human product, we have Lifeway Kefir. This one is definitely the most well-known brand for selling kefir products, yet the top 4 most predominant species in their product were not listed on their label, which I thought was very surprising. And so you can see a Streptococcus thermophilus, Lactobacillus delbrueckii, Lactobacillus paracasei, Streptococcus salivarius, all of which are pretty large abundance in this product, and none of those are listed. And I would also just like to draw attention to the end again, saying that a lot of the blue that were listed on their label was not found in the product at all. So that should be interesting. And so this is a slide to compare the animal products to the human products. So out of the 6 total products for animals, 4 of them made CFU per gram claims, and none of those products met the claims at day 1, but some did later on at 2 weeks. And then 5 out of 6 of those animal products made bacterial claims or ingredient claims, and only 1 product, which is product A, matched those species that were claimed. The genera was more likely matched, but as we know, coming down to more recent research, the effects of the species is what matters. So it doesn't really, if the genera matches, it's not as relevant as if the species are matching. For the human products, only 3 of 5 of them listed CFU per gram claims, and not one of them met them at day 1, although some did again at day 14. And then 5 out of 5 of them made bacterial claims, and all of them claimed at least 1 species that was not detected, and one of them claimed up to 7 that was not detected. And each product contained at least 2 species that were not listed by the ingredients. And I would just like to claim we do have some limitations to the study. Culture methods, like we use the MRS media, is not specific for any taxa. It's more, we were trying to cultivate as many lactic acid producing bacteria as possible, or yeast producing as possible. So with more specific assays for specific types of bacteria, we might be able to get different results for the final amount of bacteria in the product. And also our DNA extraction methods also have limitations and biases. It's an extraction kit, used to extract soil or fecal samples. So, something more specific to a dairy product could make a difference. Also, the databases get updated every, a couple times a year even. So every time it gets updated, you can run your data through and get potentially a more accurate or more detailed result. And future needs for this study is that we need to justify an expansion to testing of other kefirs and these fermented foods, seeing how much variation there is between their label and contents. There also needs more regulatory oversight on label accuracy for commercial kefir products in terms of the CFU per grams or even the specific bacteria listed. And ultimately, we will need more clinical trials to test potential health benefits for these products in humans and pets. And those are definitely lacking. So we can't make any substantial health claims just yet. And just to acknowledge, thank you to my funding, which is the USDA Hatch Grant, and to all my other co-authors, Dr. Holy, Dr. Parker, Dr. Miller, and my PI, Dr. Kelly Swanson. And of course, to the U of I and the Division of Nutrition Science at University of Illinois. Thank you. Okay.
All right, Brianna, thanks for that presentation. Super interesting. A lot of questions from the listeners. Let me go through, we'll try to answer all the questions. They're still kind of coming in. All right, so let's start. So first of all, Brianna, why do you think that the pet kefir claims were less accurate than the human ones?
I think the reason they're less accurate is due to the regulations necessary to approve human food. Those companies are held to higher standards in terms of ingredients, that they need to disclose the ingredients, more of their manufacturing process, their storage process, even transport, all of which will affect downstream results. With pet foods, those conditions are much less stringent, and so they can get away with, promoting fungi and bacteria and yeast products where, they might only have added bacterial products, but no one will really check it. So there's a lot more variation in that market for that reason.
Got it. That makes sense. All right, so we have that answered. And what do you think is the probable reason for the mismatch between the expected and actual species? I mean, are the dairy companies simply not aware of the species that they're growing, or they're not, using advanced DNA sequencing methods to check their stocks of bacteria?
Yeah, there's a lot of reasons for the discrepancy. My first answer is because these products are usually made from home fermentations and then taken to a bigger scale. Those original bacterial cultures are very specific to that environment in which the kefir grains were first cultured. And so when companies will order these like bacterial starters for kefir, for example, from another company, they might assume to have certain bacteria in them, but yeah, due to not having exposure or resources of such high accuracy like loop-seq, which can get down to the sequence level, they just don't have those resources, and so they don't have the ability to disclose the most accurate ingredient that they have in that product. I'm assuming, I would love to assume that, yeah, dairy companies will have the best interests at heart and they want to give consumers the best information. But the truth of the matter is that when you don't have that technology, you're not going to know what you really have. Or with some of these too, they might assume they are including Streptococcus thermophilus, for example, and Streptococcus salivarius is very similar. Just genome wise. So based on our databases currently, those might get categories both under Streptococcus thermophilus, but as we get more accurate databases, we can then differentiate, which maybe the companies aren't able to do right now, but they might potentially be able to do it in a year from now if they wanted to.
Got it. That makes sense. Okay. Next question. For what medical or non-medical conditions are the pet products indicated or prescribed for?
So I'm not a vet. I can't speak as a vet. I know that I've spoken to vets on this topic, and common practice is to prescribe probiotics to an animal after a round of antibiotics. And at the clinic level, I know that the vets will prescribe a very specific strain of bacteria because it's been proven to help, restart the microbiome or add diversity to a injured microbiome. But most pet consumers or pet owners will go to the pet store, see probiotic, improves gut health, and they might just buy it off the shelf, and then they might go into their vet office, and then that vet is like, oh yeah, kefir is a probiotic, sure. But the truth of the matter is that we don't have those true health administration into the health effects. We just don't know if that's the truth yet. So I think vets might just be mistaken sometimes when they will prescribe a whole product like kefir over a specific strain of bacteria.
Got it. And is there something in common for both the species that dropped out and for the unexpected species? Do you find anything in common?
When you say like dropped out, do you mean
it's species that were expected to be there but were not detected, right, and just species that were expected but you couldn't find them?
Yeah, so with a large amount, like for example, the Leuconostoc, Bifidobacterium, Enterococcus, those are advertised as being in the bottle, and we only found them in only like maybe one or two products. So the rest of them, either they're grossly mistaken or we lost the DNA, which I don't think is. I think it's more likely that they might have added it and it either did not survive, or like storaging, processing, transportation. Likely though, they thought if they buy a culture from a supplier and the supplier tells them X, Y, and Z is inside of it, they're going to trust that, whether or not the supplier, whether or not their information is accurate. So that is my best answer as to why there are so many species listed but not actually included. It could just be a supplying error or a company oversight, but they definitely need to be corrected for sure, because advertising you have bifidobacterium where there is none, or leuconostoc where there is none, can be a tricky area.
Got it. Great. And then how many sequencing reads were used per sample?
We talked about this. I believe there was around like 30,000, you were saying, or around 30,000 molecules per sample, and then a couple.
Yeah, that's exactly correct. There was anywhere between 10 and 30,000. And one thing that's important to mention here, from a technology point of view, is that is the number of short reads. Oh, sorry, of long reads that were used. So roughly, say between 10 and 30,000 individual full-length 16S molecules per sample. But then the number of short reads that were used to make those tens of thousands of long reads is in the low millions. So anywhere between 4 and 6 million short reads per sample is the number of molecules that were sequenced per sample. So 4 to 6 million short reads per sample. Next question is, what was the recovery? I guess this is kind of maybe the next few questions are more of a technology question. So what was the recovery rate of the full-length molecules for the data? Meaning how many full-length long reads were reconstructed versus how many were inputted. So the input is obviously millions or billions of cells, because this is a DNA extract, and from that very large number of molecules, we down select to a certain number of molecules, which you just mentioned is between 10 and 30,000 molecules per sample. So we barcode millions and millions of molecules per sample, but then we choose to sequence only a small subset of that, say between 10 to 30,000, because that number is more than sufficient to saturate what's called the collector's curve or the rarefaction curve. So after 10 or 30,000 reads, we're not discovering any new OTUs or ASVs. We're not discovering anything new after we're sequencing 10,000 long reads. So we barcode many more, but we only sequence as many as needed in order to cover all the biological diversity. Brianna, is there any plan to sequence the product as a metagenome as opposed to isolated colonies? Well, I guess that is what you did, right? You sequenced the metagenome, not isolated colonies.
Right. Yeah, we did the entire product. So yeah, so we
you did sequence the metagenome.
Counted everything that grew, basically.
Yeah, perfect. Next question. Was the protocol expected to capture fungi as well?
So we originally cultured all the products on Columbia blood agar as well as potato dextrose agar in addition to the MRS in hope and effort to culture those fungi. And unfortunately, under aerobic and anaerobic conditions, we did not have any fungi growing or yeast growing in those conditions. And we also did run our loop extractions through an 18S to identify potential fungi DNA, and those also came up as nothing. So even though only the first product, I believe it was product A for the pet, claimed Aspergillus fungi, we still hadn't identified none of it.
Got it. Great. Thanks for that. Next question I might be able to address. So the question is, was the full-length sequence necessary? Could the same data have been obtained with just a subregion?
I mean, I'm sure you could answer this very well too, Truval, but just from my experience using this technology and compared to other like B4 or short read sequencing, due to the long read, we were able to get a lot more accurate species level results, which is not something you can get as easily with short read. So that was the major perk, I think, with the long read versus short read.
Yeah, and that's actually an interesting question, because there was just a couple of months ago, there was a paper published in Nature Scientific Reports that basically did a head-to-head comparison between V3, V4 sequencing and loops full-length sequencing, addressing exactly the question that the listener has asked. So I can actually quickly share my screen to show you the figure specifically in that paper that addressed that question. So this is, can you see my screen?
Yes.
All right, so this is from a paper published a couple of months ago in Nature Scientific Reports, the effects of taxonomical classification by full-length 16S ribosomal RNA sequencing with synthetic long-read sequencing. This is a group, if I'm not mistaken, out of Korea that used our technology for various types of microbiomes. And if you scroll down to figure 3 here, basically they show at different taxonomical levels the effect of classifying using short read V3, V4 or long reads. And basically you can see that the effect on less resolved classification calls is not significant. Where you start seeing things kind of decouple is at the genus level, but really the most important effect is at the species level. So the species level, you can see that with this Venn diagram that they basically completely decouple, and with loop they were able to identify roughly 100 different species, while with V3, V4, roughly 74. So there was an increase of roughly 50% in the number of species that they could identify. And these additional species that were identified by full-length 16S sequencing and not by V3, V4 sequencing, those were just classified at the genus or family level for V3, V4. So it's really the ability to differentiate things at the species and sometimes strain level. And that's where really the difference between short read 16S sequencing and full-length 16S sequencing really comes about. So the last question is, were any mock communities sequenced as well?
We did sequence the Chobani yogurt as our kind of mock, I guess. That was our validation for the different methods in the sequencing. So we knew that since this was a large generic product, that those claims or CFU per gram and species taxa claims would be hopefully on the mark. So that was our mock.
Got it. Great. And maybe also to complement your answer, Brian, I can also share my screen again. There was another paper published, again, just a few weeks ago from another group. And then again, this is multiple groups on this paper. We can send out these, it's titled Accurate Microbiome Sequencing with Synthetic Long-Read Sequencing. Again, uses 16S on various types of samples. One of the sample types that they used here was a synthetic community. It was the synthetic community made by Zymo Research. What you're seeing here is figure 1 in this paper, where they're comparing the expected abundance in blue, then sequenced the same Zymo community sequenced with Zymo loop full-length 16S and then Zymo V3, V4 in black. And basically you're seeing that they all more or less match up. The synthetic communities, both the species level classification and the expected abundance match up with the expected abundances of the Zymo mock community. Okay, let me see if there are any additional questions. Okay, a few questions. Can loop pick up strain information? So the answer to that is that for some of the strains, the answer is yes, and for some of the strains, the answer is no. It really depends on the specific 16S sequences of those strains, and depends whether those strains differ in their 16S copies compared to other strains of the same species. One thing that I should mention is that one thing that we can pick up on, besides just differences in the 16S sequence of different strains, is the difference in copy numbers, in genomic copy numbers of 16S. So one of the differentiating factors between different strains of the same species is not only differences in the 16S sequence, but difference in the number of 16S copies in their genome. And for example, the classical case for this is if you're sequencing isolates. So if you're sequencing an isolate, we can easily tell whether that isolate has, say, 4 or 6 or 10 different copies of the 16S gene in their genome, and also what is the relative abundance of each one of the copies in the genome in case they are different from each other. And that is a distinguishing feature for strains that we can pick up on, but you can't pick up on that using just short-read 16S sequencing. Right. And next question is, what is the advantage of loop sequencing compared to shallow shotgun sequencing? So, Brianna, I don't know if you consider doing shallow shotgun sequencing in this case. I'm not sure that specifically in the case of low complexity samples like dairy product, it makes sense. But in more complex samples, sometimes shallow shotgun makes sense. The advantage in loop sequencing is that you'll be able to recover DNA and get species and sometimes strain level assignments from samples that have very low biomass. So with shotgun sequencing, it's very hard to get reliable results unless you have very high biomass. And the advantage with loop here is that regardless if you have a high biomass sample or a low biomass sample, you'll be able to get very deep and accurate species and strain label assignments regardless of the type of input that you have. And also in terms of cost, the sequencing cost per sample would be significantly lower with loop 16S sequencing compared to shallow shotgun sequencing. I think that the last question is, what was the parameter to classify at the species level using the Silvo 132 large subunit RNA databases? Was it over 97% nucleotide identity or over 98%? So I can answer that question. It was over 99%. So we only classify things down to the species level and if they match at least 99%. One more thing that I should mention is that within the next few weeks, we're also going to implement the Data2 workflow into Loop Genomics. So moving forward, any data that's generated with the Loop Genomics kits you can use our regular pipeline that maps the species at 99%, but you can also use the Data2 workflow that has its own algorithms for assigning either genus or species or strain level calls. Okay, I think you see there, some of the questions are just still coming in. As a follow-up to the recovery rate, if we downsample to 10 to 30,000 molecules, the kit will produce 100% of the 10 to 30,000 molecules full length? Question mark. Yes, it will. So whatever number of molecules you choose to sequence, whether it's 10,000 or 30,000 or 300,000, that is the count of the full-length molecules that you'll get. So yes, and also we only classify full-length molecules. So if there are any partial molecules where we only were able to generate, say, a V1, V6 sequence, then those molecules are not classified. We don't even attempt to classify them. We only classify full-length molecules. So all the data that Brianna just presented about with the classification results, those are all full-length V1, V9 molecules. Okay, I think that we've answered all the questions. Brianna, based on the engagement and questions from the audience, seems like everyone was super interested in your talk. Thanks so much for sharing your published paper with us. And based on what you've told me, there's another paper that's coming out in the next few days or few weeks. Can you tell us a little bit about that and how that's different from the paper that was already published?
Yeah, so by the end of this month, we're going to be having another paper published in the Journal of Journal of Dairy Science, where we just analyze the human products separate from the pet products. So we have most of the same data you saw today, just more specific to those human products and the commercial labels that they claim. And then the paper we do have published currently in the Journal of Animal Science is focusing on those pet products in particular.
Got it. Thanks.
Yeah.
Thanks for that. Okay. Tony, do you want to say a few concluding words about how the listeners can maybe get access to a recording to this presentation and how you're going to how we're going to follow up with them? Great, great. Thanks, Tony. Okay, thanks everyone for attending. Brianna, thanks again for your presentation. And we look forward to any additional questions you might have. Feel free to reach out to us. And have a great rest of your day. Bye-bye.
Thank you. Bye.